Stuart to you. He is a physiotherapist and this is his PhD Viva. It's hard to believe that he's actually standing up doing this now because it doesn't seem like 5 minutes since you were joining the team. Sam is one of the, he's an AHP, one of the first AHPs that will go through the NIHR BRU BRC funding as well, so that's fantastic to see him competing within that funding source. And he's going to talk to you today about the main findings from his work, which was exploring vision and gait in Parkinson's disease. I'm not going to say too much more. One last thing. Sam is going to give his presentation, and because this is a PhD, it's prior to his PhD Viva, we've decided not to take questions, okay? But if anyone does have any burning questions, then please feel free to send him a cheery email or come and meet him and just to address any specifics that you'd like to know a little bit more about. Okay, over to you, Sam.
Great. So, Lynn's already introduced me and discussed a little bit about what my PhD is about. So it's about vision and gait in Parkinson's disease, looking at the impact of cognition and response to gait interventions like visual cues. I'll give an outline of what I'm going to talk about today. So I'm going to talk about some background based upon a literature review which I've published. I'm going to give my PhD aims, and then I'm going to talk you through the methods that I use for my PhD and several sub-studies which I use to develop robust and accurate methods to use in my PhD. I'll then talk you through the main results of my PhD and provide a summary of those results as well. So the background of my PhD is based upon a literature review which I've published, and it's about gait impairment in Parkinson's disease. And I was interested in gait impairment in Parkinson's disease as well as the role that vision plays in gait impairment in Parkinson's, because gait impairment is a known risk factor for falls, and that's why it's of interest here. Vision and visual impairment is also a risk factor for falls as well. And there's been a developing strand of research showing that visual impairment impacts gait impairment in Parkinson's, but it's not been fully understood yet, and that's why I'm conducting this piece of research. But while looking at this relationship between vision and gait, I also needed to account for the confounding role of the cognition in gait, because robust research has shown that cognition has a role in gait impairment in Parkinson's disease, independent of demographic factors. And cognition and vision have been studied in quite separate strands of research, where individuals will do a vision and gait study, or they'll do a cognition and gait study. Very few studies, if any at all, to be honest, have looked at the relationship between cognition and vision and that impact on gait. Because vision and cognition are not as separate as they may seem from looking at the gait literature. They do have interactions between themselves among numerous levels of information processing. And those interactions between cognitive and visual processes can be termed visual cognition. And visual cognition, it's a difficult thing to measure during gait, but it can be measured through the use of saccadic fast eye movements, because of the known influence of cognitive and visual processes on saccadic eye movements. Saccades therefore provide us with a useful behavioural measure that we can measure during gait, while an individual walks, that is. And they provide the basis of our visual exploration, so that's what we look at when we walk through an environment, how much we look at things when we walk through an environment, and they likely have an impact on gait impairment in Parkinson's disease, but that again has not been looked at previously. So this forms a good theoretical model, and this review developed this model, and I used this model within my thesis as well as this presentation to talk about my results and discuss the findings of my research as well. So bear this in mind. I'll come back to this in a little while when I'm talking about my results. So the overall aims which I've covered, I've said that it's about understanding and further understanding that role of vision and gait in Parkinson's disease, while accounting for the influence that cognition has on that. And I had three primary aims in the thesis in my PhD. First was to investigate saccade frequency during gait under different attentional manipulations. So saccade frequency is how many fast eye movements you make when you walk through an environment. So how often do you look around your environment when you walk through it? And attentional manipulations that I used were environmental challenge and dual task. Again, I'll come back to these again when I talk about my protocol. And I also wanted to see the response in sarcade frequency to a commonly used gait intervention, a visual cue. Again, I'll talk about this in a little while. And finally, exploring all of the relationships between those features that were presented on that model on the previous slide there. But before I could answer the three primary aims of my PhD thesis, I had to undertake some secondary sub-studies as well. And this was to establish some robust methodology that I could utilise and base my PhD studies on, because I needed accurate and reliable data in order to present a valid argument in my PhD. So the first stage of that was to do another literature review, because one wasn't enough. So I did a second literature review, which looked at all of the Vision and Gait research that has been out published since, I forget when. But essentially I found that the majority of Vision and Gait studies use either a vision or occlusion studies, whereby an individual is placed in a light room where they can see the environment and they walk through the room and someone tracks their gait, and then they place them in a dark room where they can't see the environment and then they track their gait. Now these studies have given us some insight into the role that vision plays during gait, but taking away vision and putting someone in a dark room doesn't necessarily mean that visual processes aren't still occurring, because visual spatial information can be gathered from working memory processes and cognitive aspects. And during walking in a dark room, it places unnatural cognitive load on an individual. So therefore, we may just be seeing cognitive aspects of gait coming through there. So I reviewed all of the literature as well that looked at eye movement research, so studying how many eye movements people make during walking. And I found there's an array of methodological issues with a lot of the previous research that's looked at eye movements during gait. The first is the type of device that people use. So it ranges, I mean there's several different devices here, but it ranges from static devices during different tasks to more mobile devices, which are EOG and infrared eye trackers. There's also no gold standard for the protocol that people use. People use a variety of different things: walking, obstacle crossing, visual cues, even people do real world tasks with reaching as well. The visual metrics and outcomes that people use range from number of eye movements to latency of eye movements to fixations to just reporting general gaze data with no specific eye movements reported. And generally the sample sizes in this field of research has tended to be very small, less than 20 people per group mainly. And I was shocked to find actually once I reviewed all the literature, of the ones that use eye tracking devices, none of them reported any of the accuracy or reliability of their devices. And this is key because if you're going to base clinical decisions based on findings found with eye tracking devices, you need to know that that's an accurate device, it's reliable for what it's reporting. So that was the next step of my small sub-studies before my main PhD findings, where I investigated our decabolus mobile eye tracker, here sported by Cristiano Ronaldo here. And I investigated the accuracy and the reliability of this device in order to give me my primary outcome of sarcade frequency. So the first stage of that was to develop a novel MATLAB algorithm. This is a computer-coded algorithm that takes the raw coordinate data, which is pictured in this graph here, and provides me with the outcomes of sarcadic and fixation activity. I did this by manually going through 100 videos of individuals walking with the eye tracker, manually going through 100 videos of those eye tracking videos and picking out and counting all of the sarcades and eye movements that were utilized in those videos. I then compared my count of the eye movements to the count that the algorithm had provided me, and found that on average 85%, the algorithm will pick up 85% of the eye movements that I would pick up when manually looking at the videos. So the algorithm is accurate and can detect sarcadic activity while someone walks through an environment. So I've got a mechanism to get my primary outcome. So I took this a step further and looked at the accuracy and the reliability of my decabolus mobile eye tracker. I developed another novel novel protocol really. This has never been done before, so I had to invent a protocol in order to do this. I used a testing board, and I got people to move their eyes back and forth between two targets placed on a white board, set at 5, 10, 15 degrees horizontally and vertically. They did this while sitting, which I'll show a video of here, moving backwards and forwards there. They did it while standing as well with their head still. Again, moving backwards and forwards, a beep would go and someone would look back and forward between these two targets. And they did it while walking on a treadmill with a safety harness on as well. And they did this on 2 separate occasions, approximately a week apart. And what I did was look at the difference in the algorithm's output for the distance of the eye movements it was reporting compared to the distance on the testing board. And on average, it showed that accuracy was approximately 1 degree, and reliability, so the difference between the two sections, again was approximately on average 1 degree. So it was showing that it was an accurate, reliable device for me to use for the rest of my PhD studies. So that meant that I could use the device for my PhD protocol, which is available on clinicaltrials.gov, and it also is being published open access, so everybody can access that if they would like. And it involved 100 participants, my overall PhD. So 60 people with Parkinson's disease and 40 age-matched older adults. And they wore that Decabulus mobile eye tracker while walking in several different conditions, as seen here. So we had environmental challenge, which was straight and uncluttered environment. Then we put a doorway there. Then we put a doorway there and make them do turns left and right, distracting their attention as they walk through that area. We also did increase their attention to their walking by placing a visual cue down. These were black tape lines placed 50 centimetres apart, approximately a normal step length, for individuals to step over while completing the task. And they also did single and dual task. So again, another distraction of attention. Environmental challenge and dual task are real world things that people would come across in everyday life. And the visual cue is an intervention that we would use in the clinic to provide someone with lines to step over so that they increase their step length. For those of you who don't know what a dual task is, I'll show a little video of, so it's basically doing 2 things at once. So walking and talking, or walking and doing something else. In this case, this lady here in the foreground is walking and texting on her mobile phone. So you see her dual tasking there, walking along. She isn't very good at dual tasking, obviously, because she falls in the pond there, she's good at doing that. But the dual task in my case, for this PhD, was repetition of numbers while walking. So the individuals were tested in seated positions where they had to recall numbers back, and we took it to the maximum amount of numbers that they could recall back and got them to walk while doing that, and we tracked their eyes and their gait and everything while they were doing it as well. So again, we tracked the individuals walking by using 3D motion capture. So this is the gold standard for gait analysis, 3D motion capture. And this allowed us to track both simultaneously both eye and body movement in order to get gait and eye movement from the same task. We had these reflective markers on the head, the shoulders, the pelvis, and the feet while individuals complete these walks. And you see these are the outcomes that we get here, where we've got that little man walking through there. And that red line coming down from his head, that's his eye movement. So you can see he looks down to the ground a couple of times where he's walking there. So individuals also completed a battery of cognitive and visual function assessments. So ranging from things that we do in the clinic, from Mocker assessments, Addenbrooke's, content sensitivity visual acuity charts that are commonly used, to more complex assessments of attention, from which we derive our power of attention and fluctuation of attention measures. I had several visual spatial outcomes from the JLO to the clocks 1 and 2, and digit span for working memory as well. Like I said, we had them sit down and repeat those numbers. So I'm now going to talk about the results. I'm going to do it in 2 stages. I'm going to start with the descriptive data about the impairments in different aspects, and then I'll go back to the model and talk about the relationship between these aspects. So not unsurprisingly, cognition and visual function in Parkinson's disease were impaired compared to the age-matched older adults. Similarly, gait was impaired in Parkinson's disease. Individuals during the gait tasks had shorter steps, they had longer time spent with both feet on the ground, they walked slower. These things were influenced by the environmental challenge and the dual task, whereby they got slower and had shorter step length if they had a doorway to turn through or if they had a dual task to do. So gait was impaired in Parkinson's disease. What about eye movements in Parkinson's? Well, this is just generally the eye movements of a healthy control completing one of these walking tasks. And we see that they're making quite rapid, fast eye movements as they complete this task. Several different small ones, large ones, fast ones there at the end. And this is a Parkinson's disease participant performing the same exact task. And we see they're very slow to react, very slow eye movements, not very big eye movements, not very many of them compared to the control. So generally they had eye movement impairments, non-significant for the majority of these aspects, but they were impaired. But what about the primary outcome of saccade frequency during gait? Well, this is a control participant walking straight in an uncluttered environment. And we see this control looks down to the ground several times as they complete this task, looking at relevant areas, the ground where they're going to walk. And then we see a Parkinson's participant perform exactly the same task. And we see that they don't really look at very much in the environment at all. They just look straight ahead of the wall, aren't really looking at the floor where they're going to be walking or anything relevant to the task. And this obviously has connotations for trips and falls. If you're not looking at things where you're going, places where you're going to walk, you may trip over something and fall. And it's also similar to previous work by Gallner and Vittorio in 2012. But those previous studies have demonstrated that there were non-significant findings during straight walking in uncluttered environments, whereas my studies demonstrated significant differences and significant reduction in saccade frequency in an uncluttered environment. And that's likely due to the robust methodology as well as the larger sample size that I've taken this from as well. But what happens if I intervene and give someone a visual cue? Well, this is a control participant performing this task with a visual cue. We see again, they look down at the ground several times and make a few more eye movements as they come through that environment. And then a Parkinson's participant. And remember, the Parkinson's participant was just really looking straight ahead before. And now if we give them a visual cue, they're looking down at the ground or where they're going to walk, where they're going to step as they complete this task. So they're making more eye movements and they're attending to their gait and the task that they're doing. And these results that I've just discussed are reflected in these graphs here, where people with Parkinson's disease made less eye movements than the controls during all the gait tasks. But they were able to increase their eye movements if they had to make a turn. So if they had to make a turn, they did increase their eye movements, but always stayed below the controls. Similarly to controls, if they were under dual tasks, so if their attention was distracted, they made less eye movements than under single task. And particularly so in Parkinson's disease, you see the gap between the single and dual is a lot larger in Parkinson's. But what happens if we give them an attentional intervention, a visual cue? As we saw before, the eye movement frequency increases in both groups, under single and dual task, but more so in Parkinson's again. And we see that, interestingly, under dual task, when you use a visual cue, saccade frequency is actually maintained. It's similar to your single task. So they're looking a lot more often, they're looking as often as they would if it was just they weren't being distracted by a secondary task. And even more interestingly than that, both groups actually got better at their dual task. So they maintained the amount of exploration they were doing, and they also were able to do better on a secondary task while performing this with a visual cue. And this demonstrates that attention likely has a big role here to play, because it's freeing up resources, this visual cue, this attentional visual cue. So like I said, I'm going to use my model now to talk about these relationships. So I've demonstrated that all of these characteristics, cognition, visual function, saccade frequency, and gait, are impaired in Parkinson's. But now I'm going to talk about the relationship between these features. I'm going to start at the top of the model and work my way down. I'm going to discuss all of them. So the first aspect is the relationship between cognition and visual function. And what I found was that in both Parkinson's participants and older adults, there were significant associations between these features, forming that visual cognitive aspect. More so in Parkinson's disease, particularly for the power of attention fluctuation with attention measures and contrast sensitivity. But generally both of these groups had these visual cognitive aspects. So the next stage of the model is then to look at the relationship between cognition and saccade frequency, visual and function and saccade frequency, as well as demographics. So I looked at associations between these features, and surprisingly, actually, there wasn't a lot of associations in these features. Very little consistent association, particularly. In Parkinson's disease, visual function actually didn't relate to saccade frequency during any of those. Only in the single task in healthy older adults was contrast sensitivity related to saccade frequency. And the only consistent findings were for attention in Parkinson's disease, which demonstrated that those who had better attention actually increased their saccade frequency more during gait than those with poorer attention when they were presented with an environmental challenge or a visual cue. And similarly, there was a consistent finding, an unusual consistent finding for motor severity. So motor severity had an impact on saccade frequency as well. And that's unusual because that's actually the opposite of that's been found in the literature, where Tim Anderson's group and Roger Barker as well have done some research looking at motor severity and saccade latencies and found that there's no relationship there. So I wanted to investigate this further and take it to the next stage after these correlations. So I looked at multiple regression analysis. So I'm back to the model, and I did multiple regression analysis in 4 separate steps. Step 1 was demographics. Step 2 was cognition, which was controlled for demographics with. Visual function, again controlling for demographics. And then visual cognition in the middle, which is the accumulation of all 3 of the others. And what I found was that under single task in Parkinson's disease, attention and contrast sensitivity were independently related to saccade frequency. But there were some interesting findings in the other steps as well. So contrast sensitivity was only related to saccade frequency once it was entered into the step with cognition, showing there may be some relationship there that then drives the relationship between visual function and saccade frequency. Motor severity did still come out in the separate demographic and visual function models, but when entered with cognition, it was not present in the not as prominent, showing that motor severity may be mediated by cognitive elements. And along those lines, when under dual task, motor severity came out in every model. So once we took attention away, you see motor severity come to the foreground and become more prominent. So attention is mediating this response, and again, attention plays a big role in saccade frequency here. So then going back to the model again, the next element of the model is to look at saccade frequency and gait and the relationship here. So gait, these are all the gait characteristics that we were able to get from the 3D motion capture. And interestingly, for people with Parkinson's disease, there wasn't a significant correlation between saccade frequency and gait. Only for controls during gait was step length and velocity related, whereby if you made more eye movements and explored more, you were faster or you had longer steps. But when you manipulated attention with a visual cue, that's when we saw relationships between people with Parkinson's saccade frequency and their gait characteristics. Whereby, again, if you made more eye movements, you walked faster and had better gait during these tasks. And this again shows that attention has a role here again, because when we manipulate it, we start to see outcomes. So the final stage of this was to enter the model as a whole, to put all of the relationships between these features into a model, looking at a structural equation model. I did this. So I entered all the hypothesized links between these features into a model and looked at the direct and indirect relationships. Direct relationships being where one variable, so say cognition, influences gait. Indirect relationships being where one variable may influence another variable, but it goes through a third variable. Okay? So this is what I found for that. So interestingly, visual function didn't have direct relationship to either saccade frequency or gait within this model. And saccade frequency didn't have direct relationship to gait either. But cognition had direct relationship to all of the other features. And visual function and sarcade frequency, once these hypothesized relationships had been put in with cognition, we saw indirect links, whereby visual function went through cognition to influence sarcade frequency. So if you had poor vision, you likely had poor attention, and that led to poor sarcade frequency, or poor gait. And sarcade frequency was the same, whereby sarcade frequency had indirect impact on gait in Parkinson's disease through attention. So again, attention being central to all of the relationships within the model. And just to summarize all of those results, initially I'd show that you can use an eye tracker to measure sarcades during gait in older adults and people with Parkinson's. But main results were that people with Parkinson's don't explore their environment as much as controls, particularly when they get distracted. And this obviously has connotations for tripping over things, falling, if you're doing two things at once. They can increase their sarcade frequency in response to environmental challenges, and they do the same as controls in reducing their sarcade frequency with a dual task, but more so in Parkinson's again. And interventions like visual cues can actually increase the amount of exploration in sarcade frequency an individual, older adult or person with Parkinson's does, and it's maintained under dual task, particularly in Parkinson's disease. But attention plays a central role in these features. In visual cognitive function does influence gait impairment, but only through attention. So therefore, if you've got impaired cognition, particularly attention, it's likely to impact both your exploration of your environment as well as your ability to walk through that environment, which again could lead to trips and falls. And I'd just like to say thanks for listening, and thanks to my supervisors, Professor Lynn Rochester, Dr. Sulo, Dr. Burgalna, the rest of the Brain and Movements Research Group. And again, if you want to contact me, my email's on there.